Assalamu alaikum. Today we will be going to do a little bit about Staphylococcus aureus. This would be a presentation that would be including only the basics of Staphylococcus aureus. We'll start from the beginning, and then I will take you to the next levels in the next videos. Now we will only be doing the very basics, which is related to Staphylococcus aureus. So what is Staphylococcus? It is a gram-positive coccus, and it is a facultative anaerobe, non-motile, non-spore forming, and since in the gram stain it looks like a bunch of grapes, so they do look like branches of grapes while they are done in the gram stain. Inshallah, I will do the gram stain later on in the next videos. Today we will only be doing this basics about Staphylococcus. So we should know the virulence factors or the pathogenesis. So first, for that pathogenesis, we should be able to know about the virulence factors of the organism. So first of all, what are the virulence factors? They say there is the expression of adhesins needed for colonization. There are certain adhesins which they express, and what do? They are used for inoculation and local colonization. Organism cannot enter the intact skin or the mucous membrane. It is required. There is a break in the integrity of the skin is required. Okay, so it helps it to colonize the site. Okay, it also helps in the nasal colonization of the organisms. Then there are certain toxins which include the following: the heat stable toxin, the enterotoxin, the toxic shock syndrome toxin, which is also called a superantigen. Then there is an exfoliative toxin, and then there is an alpha and beta and delta gamma. There are four kinds of these toxins, and then there is a PVL, Pantone Valentine leukocidin toxin is also there. So a few things I will tell you about these little bit of these toxins. For example, the heat stable toxin, it is associated with pseudomembranous colitis, whereas the toxic shock syndrome, you know that it occurs in the people, the females who use tampon, or the when the person who has nasal bleed and certain packing is placed there, so it will cause a toxic shock syndrome. The alpha toxin is basically, it disrupts the smooth muscles in the blood vessels. The beta toxin is a heat labile sphingomyelinase, whereas the delta toxin is a cytolytic. It is cytolytic to erythrocytes, while as the gamma toxin, it is produced by cell strain, and it's produced by certain cell strains, not all of them. And we are very familiar with PVL, Pantone Valentine leukocidin, which is a lysogenic cytotoxin. It causes necrotizing lesions such as necrotizing pneumonia. Okay. Then we have enzymes. The enzymes are catalase, which neutralizes H2O2, uh, which is harmful for the bacteria actually. Then the coagulase, it helps the organisms in the abscess formation. It also helps in the walling off phenomena. It retards neutrophilic migration and inhibits phagocytosis. It also helps in the formation of biofilm. Then there are certain enzymes which include DNases, proteases, lipases, staphylokinases, hyaluronidases, penicillinases, and beta-lactams. We are familiar with penicillinases and lactamases, which of course help in the destruction of the antibiotics, and they play a very good, a pivotal role in the antibiotics, in the resistance of the antibiotics by this organism. Hyaluronidase is basically also called a spreading factor, whereas staphylokinase is also it helps in fibrinolysis, and it also helps in bacterial spreading. Whereas DNase, protease, and lipase, they break down these products, that is the DNA and the proteins and the lipids. They provide nutrition to the organism. Then we move on towards the hemolysins. Hemolysin production is important because it hemolyzes the RBCs to get iron for energy. There are two types of hemolysins, alpha hemolysin and gamma hemolysin. They are also helpful in the pore formation, and this helps in the destabilization of the membrane. Then there are certain pigments which are called staphyloxanthins, which are also which are also the antioxidants, and they impair certain neutrophilic functions. Then there are certain cell wall components which include protein A, teichoic acid, lipoteichoic acid, peptidoglycans, polysaccharide capsules, and surface receptors for specific staphylococcal bacteriophages. So protein A is it helps in the binding of the Fc portion of the IgG at the complement binding site. ठीक है, the teichoic acid helps in the adherence. The lipoteichoic acid is that the cytokine, it helps in the release of the cytokine from the macrophages. The peptidoglycans is the endotoxin, whereas the polysaccharide capsule is mostly anti-phagocytic for the bacteria. It helps the bacteria to survive basically. So the infections caused by the staphylococcus are two types, suppurative and toxin mediated. The suppurative are local and disseminated. The local may include the skin and wound infections like folliculitis, peduncles, carbuncles, cellulitis, and bullous impetigo. Apart from that, it also causes conjunctivitis in for the eyes, mastitis in the breast tissues, and lymphangitis. Then there is a disseminated disease which is sepsis, bacteremia, endocarditis, pneumonia, septic arthritis, and metastatic abscess formation. The toxin-mediated part of the diseases which are caused by the staphylococcus are basically, of course, they are mediated by the toxins. They include the toxic shock syndrome, the enterotoxin also is there, and then there is exfoliative toxin which causes scalded skin syndrome. Then there is toxic epidermal necrolysin, and then there is pemphigus neonatorum. These are all the diseases which is caused by the toxins of the staphylococcus. The PYR means pyrrolidonyl arylamidase, PYR. It is positive in Staphylococcus epidermidis, Staphylococcus lugdunensis, and Schleiferi, Staphylococcus pseudointermedius, Staphylococcus intermedius, and Staphylococcus hemolyticus also. ठीक है, it is negative for the Staphylococcus aureus, whereas the Staphylococcus aureus, the slight coagulase is positive and the tube coagulase is both positive in the Staphylococcus aureus infections. It is organisms. The difference between Staphylococcus aureus and Staphylococcus lugdunensis or Schleiferi we can do is by knowing that Staphylococcus aureus is PYR negative and ODC negative, whereas Staphylococcus lugdunensis is both these tests positive. How will we differentiate between Staphylococcus aureus and intermedius? Again, the PYR is negative in Staphylococcus aureus and VP is positive, whereas in Staphylococcus intermedius, PYR is positive and VP is negative. Next, we will go to the, this was just to identify the Staphylococcus aureus and differentiate it from the other organisms. We can also put an API staph, a white tech, a microscan, etc. For these are the a bit highly specialized tests which can be done in the laboratory for the identification of the Staphylococcus aureus. Apart from this, we can do serology for toxin detection also, and there are two ways. There is a lattice agglutination test and immunochromatography. Then, if the organism turns out to be MRSA, methicillin resistant Staphylococcus aureus, we can do expert MRSA or MRSA, and which is the turnaround time for which is almost 1 hour. We can also do the typing methods that we can use is I will just give the abbreviation and later on I will give you the detail of these. The first one is PFGE, the next one is MLST, the next is MLVA, and then there is ribotyping and MALDI-TOF MS. So the word PFGE stands for pulse field gel electrophoresis. MLST stands for multi-locus sequence typing. Then there is MLVA, the word that I said, that was multi-locus variable number tandem repeats analysis. Then there I said MALDI-TOF MS, matrix assisted laser desorption, time of flight, mass spectrometry. This is how we will be able to do the this typing. These are certain typing methods that can be used to identify Staphylococcus aureus. Then there are a few other points that needs to be known, and then that there are certain small colony variants of Staphylococcus aureus that can emerge on these agar plates. So they result from the limited nutrients and selective pressure. They cause Clinically they cause persistent relapsing infection. They persist intracellularly, and they rapidly switch between the small colony variants and the normal population. And switching is can be both phenotypic and molecular. Apart from this, this is the extra information that I wanted to give you. Apart from this, we have certain gene experts and gene experts for MRSA detection also. We have fish techniques, and then there is electrospray ionization, mass spectrometry, and mild of MS that I told you before for the detection of Staphylococcus aureus. Then there is a very every time a repeated question which is asked with that, what is the difference between Staphylococcus and Micrococcus? So there are certain differences that are shown on the slide. Apart from these differences which are shown on the slide, it should be noted that the Staphylococcus aureus is a facultative anaerobe, whereas Micrococcus is aerobic. ठीक हो गया. And then there is it is the Staphylococcus aureus is sensitive to antibiotic furazolidone, whereas the Micrococcus is resistant to the antibiotic furazolidone. These were 2 points that were not written in this slide, which I just told you. So the detection of MRSA, we will go through it very quickly. MRSA is methicillin-resistant Staphylococcus aureus. The details about it, the clinical findings, and other things will be I will upload later in the videos. There are 4 methods for detection, which is culture-based method, the immunoassays, the molecular methods, and the antibiotic susceptibility test methods. So the culture-based methods, as the name indicates, they include the cultures, and they include the culture plates. So the mannitol salt agar with 7% NaCl can be used. The chrome agar MRSA can be used. Then there is a broth enrichment with 7% NaCl can be used before culturing for the MRSA. The semi-selective TSP with 2.5% cefoxitin and astronam can be used for the detection of MRSA. Immunoassays mostly includes that can be done on the colony, and they detect the PBP2A. Whereas the molecular methods include the reverse transcription PCR, the multiplex PCR, the gene expert. Then their question may arise that what kind of molecular test can be done on the swabs. So the molecular tests that can be done on the swabs are Cepheid, Expert MRSA, Max MRSA. MEC-A and MEC-C gene can also be detected, and export MRSA, MRSA, basically MRSA no, MRSA can be detected from, these are the tests that can detect MRSA from the swabs. Whereas the question can come that how can an MRSA be detected directly from the blood in case of sepsis, for example. So we can detect it by a PCR, by a PNA fish, Redica, Septifast, and T2 step. These are just for the for your quick summary kind of thing for MRSA. So the AST methods includes 2 types of discs that are used for the these antibiotic susceptibility test methods, and they are the cefoxitin and oxacillin. The cefoxitin disc can be used. The cefoxitin disc can be used for the disc diffusion method, where a 30 microgram disc is used, whereas for the broth microdilution we can use 4 microgram per ml of cefoxitin for the detection of methicillin-resistant Staphylococcus. There are multiple species that can be detected by these. There are certain species that can be there are more we can हम उनको बेहतर तरीके से detect कर सकते हैं इस methods के through जो कि methicillin resistant होंगी. So there are oxacillin is also a second disc that can be used, and for that we can use 3 types of methods can be used. The disc diffusion method is for the Staphylococcus species, and it does not include Staphylococcus aureus, whereas a broth microdilution can be used both for detection of methicillin-resistant Staphylococcus aureus and methicillin-resistant Staphylococcus species. Whereas the oxacillin salt agar is only used for the Staphylococcus aureus, which is methicillin resistant, and any growth it uses 6 microgram per ml of oxacillin, and now on a Mueller-Hinton agar with 4% NaCl, any growth on that agar will be considered at the growth of methicillin resistant Staphylococcus aureus. So these were a few things that were for the Staphylococcus that I wanted to go over. For the next, like for the vancomycin resistant Staphylococcus and even the MRSA, the methicillin resistant Staphylococcus aureus, the detail about their tests and the detail about their diseases they cause and how they are detected will be inshallah uploaded in the next video. For the time being, thank you so much. Allah Hafiz.